Hey everybody, welcome back to another AP Biology video. In this video, we are going to cover AP Biology topic 3.5, which handles the process of photosynthesis. Now, this correlates to the Biology in Focus textbook chapter 9, I believe. It's either 8 or 9. One of them is cellular respiration, and I always get them mixed up. But we're going to cover a lot of details in this topic. This is a process that can be as simply described or as complicatedly described as we really want to get into it. So I'm going to try and run somewhere in the middle. Now, before we get into any of this content, what I think it's important for me to communicate to you is that I'm going to go through a lot of processes, a lot of things that have multiple steps, a lot of things that have a lot of working parts. And as far as the AP exam is concerned, we do not need all of that detail because we want to really understand how something works. And in order to understand how something works, we have to get into those details. The AP exam is focused more on the general activity that these processes are providing to different cells. So as we go through this, I think it's important that you keep in mind that you do not have to memorize any of these processes. You should know what's going on. You should recognize when different activities are taking place. But memorizing the steps of, say, oxidative phosphorylation when we get to cellular respiration is not the goal here. So let's just start really simply with a basic rundown of photosynthesis. As we established in Unit 8, when we talked about food webs and energy flow through our ecosystems, all of the energy that we have to use here on Earth comes from one source, the sun. And the only organisms that are able to harness solar energy and convert it into a form that organisms can use in order to do work are photoautotrophs. So photo, that prefix meaning light, auto meaning self, and troph referring to feeding. So this is feeding one's self through the use of light. So the process that photoautotrophs use in order to take solar energy and convert it to a form that they can use to generate cellular energy is photosynthesis. Now, photosynthesis overall is what we term an anabolic process. So anabolism referring to us being able to build molecules. And we're building these sugar molecules primarily through energy that they're capturing from the sun. Now, the more simple equation for photosynthesis is the one down here in the blue. And this is a general overview. This is taking out all of the different parts that are coming in and out and that are being recycled throughout and getting it down to the nitty-gritty. So if you think about the things that plants need to survive, they need water, they need carbon dioxide, okay? These two reactants enter into this process in different stages. So you can see I've broken here down the light reactions and the Calvin cycle, which we'll get into in a little bit more depth further on in this video. But they come in and they're used in different ways in order for us to create essentially the building blocks of a sugar molecule. We don't get glucose straight out. We get some intermediaries and we release oxygen gas as a byproduct. And oxygen gas is going to come back when we look at cellular respiration as a very important molecule for these processes to take place. So let's zoom out for a moment and get to know the organelle in which this whole series of stages takes place, and that is the chloroplast. If you recall from our discussion on organelles and cell structure, that cells compartmentalize functions in order to increase efficiency. So we get a lot of folding in these organelles, which increases our surface area without limiting our volume too much. And so chloroplasts are one of those unique organelles that, because they're very, very similar to some free living photosynthetic bacteria, it is very likely that these are the result of endosymbiosis. We also discussed endosymbiosis in the previous unit, where we looked at the structure and function of our chloroplasts and identified a couple of things that lead us to the conclusion that they were engulfed by a proto-eukaryotic cell, and they eventually just became part of the cell, and the cell uses them in order to garner all of this energy. So you can see here that we have a lot of structural similarities between our chloroplasts that we find in photoautotrophic eukaryotic cells and that of a free-living cyanobacterium. Now, there are some differences, of course, over a long period of time. Structures change, and that is essentially just the process of evolution in a nutshell. But what's important is that all of the materials and enzymes necessary for your plant cells to engage in photosynthesis are also found in other free-living cells. So together, these photoautotrophs have very similar structures that allow them to complete this process. Now, chloroplasts are not found in all plant cells. In fact, we really only find chloroplasts in areas of the plant where we would expect sunlight to be easily accessible. So we are going to really look most closely at leaves. Leaves are our major locations of photosynthesis, and therefore they are chock full of chloroplasts in their mesophyll cells. And when we look at the structure of a leaf, we have these rows of cells that are grouped and have all of these chloroplasts that allow them to capture light energy. And then on the underside of our leaf, we have these pores, these stomata. These are regulated by guard cells, and they can open and close to allow gases to exchange into the tissue from the environment. So when we discuss our gases, in this case, we're talking about carbon dioxide, which is a reactant, and oxygen gas, which is a product. These are coming and going through the stomatal pores of our cells. So looking at the structure of our leaf, we can see how this process is designed to function. We have areas where we can gain light energy, areas where we can absorb carbon dioxide and release oxygen gas. And so the structure here lends itself to the function that plants have to engage in photosynthesis. In the previous unit, we looked at the structure of our chloroplast, but I do want to highlight it once again because the structure determines how the chloroplast functions. So you'll see here that the chloroplast actually has two outer membranes. This essentially leads us to the conclusion that it was engulfed twice. And so the outer membrane is more like that of the cell's outer membrane. And then the most interior membrane is highly folded, and it's highly folded into these stacks, which we call granum. Granum is a Latin word that means stack of coins, because the granum and each one of those little cylinder discs, those are called thylakoids, and the thylakoids are stacked and they're interwoven within the chloroplast structure to allow an increase in surface area. An increase in surface area means that we can create more membrane-bound processes. So the first process in photosynthesis that we're going to look at really relies on these membrane-bound proteins in order for it to function. So we have a lot of membranes for that process. And then outside of the thylakoid, we have this fluid, which is termed the stroma. And the stroma is where we're going to get our Calvin cycle. It is the portion of our process that is cyclical. We're building things in the stroma. And we really don't need those membrane-bound proteins to work because everything's done through enzymes. So, knowing the structure of our chloroplasts will be immensely important in identifying where these different reactions are taking place, but also understanding the general pathway by which our molecules will move. So photosynthesis, as an anabolic reaction, is also a redox reaction. So we have coupled reduction and oxidation reactions. So keep that in mind when we talk about these energy carriers. They're going to be carrying electrons and therefore will be either reduced or oxidized throughout this process. So we are oxidizing oxygen. We are removing its hydrogen in this process. We are removing its electron, and we are reducing carbon dioxide in two major processes. The first process is the light reaction, and this can also be termed photooxidation. Sorry, not photooxidation, photophosphorylation. So this is the photo part of photosynthesis. This is where we are going to take in solar energy and we're going to create chemical energy carriers. We're going to latch electrons into these energy carriers so that they can take all of that energy and move it on to bigger and better processes to build us sugars in the Calvin cycle, also referred to as the carbon fixation reactions, and even less commonly referred to as the dark reaction or light independent reaction. You know biology, we love to have 6 different names for everything. And so it's during the Calvin cycle where we're producing the sugar that is the eventual product of this process. So this is the synthesis part. We are building things in the Calvin cycle. And we're going to talk about each one of these in great detail, but it's important to understand where this energy is coming from. And because the energy driving this process is light, let's review a little bit about light as an energy source. You hopefully recall from previous science classes that light or radiant energy is a form of electromagnetic radiation. It's a series of oscillating waves, electrical waves and magnetic waves, and these are oscillating in different directions. And so light energy is transmitted through these waves. Now, waves that are electromagnetic, they all move at the same speed, the speed of light, but they differ in their wavelength and their frequency, which are inversely related. So as we move into shorter wavelengths, as we go from red to violet, we increase the amount of energy that that wave is carrying. And so different pigments, different chemical molecules within the chloroplast, they're all designed to react differently to different wavelengths of light. So when we look at the chloroplast of a photoautotrope, they're going to contain a wide variety of pigments that allow them to harness different amounts of energy through different wavelengths of light. Three of the most commonly found pigments within photoautotrophic cells are chlorophyll, and chlorophyll has two forms, and some accessory pigments known as carotenoids. I like to think of carrots when I think of carotenoids because they kind of have that yellowy orange tone to them that is what is reflected. So you can see here the different wavelengths of light that these pigments will absorb, and they're not all on top of each other. This is important because it allows your cells, the plant cells, to absorb a wide range of light. We don't want to put all of our eggs into a single basket when we can spread it out and allow us to get a wide range of different colors of light and aid in the process of gathering the light for our needs. So you guys probably know that leaves are green because green is reflected, and that is what is entering our eyes. But chlorophyll has to absorb the opposite in this case. So we're absorbing primarily red light and violet blue light, and we get different ranges therein from our different pigments. So the graph that I have here is what is known as an absorption spectrum. So an absorption spectrum plots the different absorption wavelengths of light versus our pigments. So we have optimal ranges of wavelength that these pigments work best in. Chlorophyll A does the heavy lifting in this case, hitting really a good range of light, but we also have lesser chlorophyll B and our carotenoids to pick up the slack. Now we can take all of the different absorptions developed by these different pigments, and we can look at their ability on an overall sum scale. And so that is where we get what we call an action spectrum. So this spectrum is the activity or rate of photosynthesis versus our wavelength. So it is all of the pigments put together onto one graph. And so that's how we get a single curve. But you'll see here that photosynthesis operates best in that blue-violet range and that red range of wavelengths. So we're not getting a whole lot of activity in the green wavelengths. Now, this is an evolutionary benefit to our plants because this is going to get us the most energy in the violet range, and we're getting the other end of our spectrum as well. So now that we've got all of our background under our belts, we are ready to get into the process. So embedded within the thylakoid membrane, we have these light harvesting complexes. And these complexes are made up of a reaction center, and the multiple reaction centers form what we call a photosystem. So photosystems are the different complexes of materials and reactants that are embedded within our thylakoid membrane to absorb all that great light energy. Primary activity of our photosystem is to essentially hold on to an unexcited electron. And through a series of kind of passing the buck, the light energy in the form of a photon is absorbed by that complex, and it excites our electron. So it increases the potential energy of our electron by moving it to a higher excited state. And so it is this potential energy that we will utilize in order to move our electron along and use the kinetic energy of its movement in order to do other things. So when we look at these photosystems, and I am going to go in a much more simplistic diagram for the next little bit. If we look at our thylakoid membrane, we have two photosystems that are embedded. And so we have photosystem 2, which is the first photosystem. They're named based on the order in which they were discovered. So photosystem 2 is first, and this absorbs light energy best at 680 nanometers. And then we have photosystem 1, which absorbs light energy at around 700 nanometers, and it acts second in our process. Now, these embedded photosystems are in addition to what we term ATP synthase, which is an embedded protein which contains an enzyme that allows us to phosphorylate ADP molecules and produce ATP, or cellular energy. So there are actually two versions of photophosphorylation that are involved in the light reaction. There is non-cyclic, and then there is cyclic. We're going to start here with non-cyclic because this is really the heavy hitter out of the two. This is going to involve a linear flow of electrons. The electrons are not being cycled, therefore non-cyclic. And we are using light energy, photo, to phosphorylate or add phosphate groups to ADP to form ATP. So that's where we get photophosphorylation. So we are also going to produce, in addition to ATP, our electron carrier, NADPH. And so all of this is using light energy. So if you guys are ready, strap in and we will go through this process. So we start with light energy being absorbed by photosystem 2 at around 680 nanometers, and this causes our electron to be excited. So let's get some light energy in here and excite our electron. It is going to move to an excited state. Now our electron is activated. So the electron that we lose, we can replenish it by oxidizing water. So we take in a water molecule and we take its electron and we produce a couple of hydrogen ions, and then for each water molecule we get half of an oxygen, a diatomic oxygen molecule. So we actually need 2 water molecules in order to produce a full molecule of oxygen gas. So obviously these are happening in quick succession in many places within the membrane. So this is really just extra detail. So now that we have this excited electron and our previous electron is replenished, our electron is going to pass through an electron transport chain, and the kinetic energy of its movement is going to allow us to push hydrogen ions into the lumen of our thylakoid here. So we move our electron, and that allows for hydrogen ions to move into the lumen of our thylakoid. So we are moving them from a region of low concentration to an area of higher concentration eventually. So this is going to require energy, and the energy source in this case is the movement of our electron. Now you'll notice that now that our electron has fallen back down to a grounded state, it needs to be excited again. And so this is where photosystem 1 comes into play. We get new light energy to come into photosystem 1. Remember, photosystem 1 operates around 700 nanometers, and this is going to excite our electron once more. So light energy comes in, excites our electron, moving it from its grounded state, and we are ready to let it move once more. So now that we have our excited electron, we're actually going to pass that excited electron onto an energy carrier. So our electron carrier in this case is NADP+. So it has a positive charge, and once it picks up an electron, it is going to become NADPH. So it is just here to kind of ferry or taxi our electron from the light reaction to the light independent reaction, the Calvin cycle, the carbon fixation reaction, whatever name we want to give it today. And you can think of this like a little energy battery. So we are adding our electron to our NADP+. We are reducing that molecule and we are producing NADPH. So now that we have this buildup of hydrogen ions on one side of our thylakoid membrane, so now we have a higher concentration of hydrogen ions on the interior and a low concentration on the exterior, we have formed an electrochemical gradient, and we can use that electrochemical gradient to do work. So hydrogen ions are going to flow from their region of higher concentration to the region of lower concentration outside of the thylakoid membrane. And the movement of our hydrogen ions, think like a dam, okay, for harnessing water energy through its flow. The flow of our hydrogen ions is actually going to spin a little rotor in our ATP synthase, and that is going to allow us to phosphorylate our ADP molecules and create ATP. So this is the process of non-cyclic photophosphorylation, otherwise known as the light reaction. So we are taking in solar energy, we are using that solar energy to create an electrochemical gradient with our hydrogen ions, and we are latching our electrons into an electron carrier. Now, both the products, NADPH and ATP, will go on to our Calvin cycle, where they will drop off the things that they've picked up, and we'll use those to do work. So one of my favorite things to show off is what ATP synthase actually looks like in the cell. It is a globular protein that contains an enzyme by which phosphorylation can occur. And we term the process by which ATP synthase works as chemiosmosis. So chemiosmosis because we are moving our molecules and using those molecules to generate the energy necessary for this enzyme to phosphorylate our ADP molecules. So the unfortunate thing is that plant cells don't often create enough ATP for use in the Calvin cycle. The Calvin cycle requires a total of 18 ATP molecules in order for it to function optimally. So we have a secondary version of photophosphorylation known as cyclic photophosphorylation that helps us to produce extra ATP molecules, but without creating additional NADPH. So cyclic photophosphorylation does not require both of our photosystems. It only requires photosystem 1, and so it operates in a slightly different way. So this is what we will look at next. So first things first, Light energy, again, we're getting our light waves in around 700 nanometers, and that is going to come in and excite our electron. So we're going to let light in. It is going to take our electron from a ground state to an excited state, and our electron is then going to cycle or circle back through the thylakoid. And so we're pushing our hydrogen ions through into the lumen as our electron cycles through. And we create that really beautiful cycle of our electrons. So this will continue until we have enough hydrogen ions on the interior of our thylakoid. And once we have that higher concentration, we can move those hydrogen ions, just like before, back outside of the thylakoid into the stroma and produce ATP. So I want you to take some time to really familiarize yourself with these two processes involved in photophosphorylation, because these will allow us to really understand how our plant cells and other photoautotrophs harness light energy and create the materials that they will need to form sugars. We want to know the differences between these and the adaptations that they provide to our photoautotrophic cells. But we are not quite finished with photosynthesis. We're only about halfway through. The next piece that we are going to look at will be the Calvin cycle, where we are going to take the NADPH and the ATP produced from our light reactions and use it to make sugar. So the products of our light reaction are what are going to move on to our carbon fixation reaction, also known as the Calvin cycle, also known as the light independent reaction. And they are going to be used within the stroma in order to fix carbon dioxide and convert it to the building blocks of glucose. And this cycle takes place in 3 stages. We have carbon fixation, we have reduction, and we have regeneration. Now, it's important to note that the cycle itself has to occur twice in order to produce a full molecule of glucose. And so we can have multiple cycles going on at the same time within the stroma in order to produce glucose at an optimal rate. But it is important to keep in mind because different diagrams of this process will describe it differently. So let us look at this process as a whole. First, we're going to have carbon dioxide. Carbon dioxide is going to enter into the leaf cells through the stomata, and there within the mesophyll cells, our carbon dioxide will combine with RUBP, which is a carbon-based molecule that this process catalyzed. The enzyme Rubisco is going to form us a 6-carbon compound. So we're going to have carbon dioxide come in, form with RUBP to produce this 6-carbon compound. So we're fixing our carbon. Now this molecule is relatively unstable, so it rapidly is broken down into 2 molecules of 3-phosphoglyceric acid, or PGA. So we have 2 PGA produced from this. Now's where we get into those NADPH that are carrying our electrons from the light dependent reaction. So using energy from the reduction of ATP to ADP and NADPH to NADP+, our PGA is going to be converted into G3P. So here we have the reduction of our energy carriers. And once those are reduced, they can return back to the light-dependent reaction where they can pick up more energy. So the G3P that is produced from this, so we have a couple of different pathways that it can take. So one of them we're going to drop off, and that is our building block for glucose. And the other is going to re-enter our cycle so that we can replenish our RUBP. So that is going to involve the reduction of more ATP in order for us to get back the original RUBP molecule that we had to start with. So through this entire process, we are converting our carbon dioxide to a different form that we can go on to use to build sugars. So as I stated, again at the top, this is going to require our cycle to complete twice, so we can have multiple cycles going on at the same time in order to make this process a little bit speedier and allow our plant cells to create all that great sugar that we need in order to derive cellular energy. And with that, that is where I'm going to leave you guys for today. So keep in mind that there are a lot of details, and I left out a good deal of the detail in this case. We should recognize the activity of our ATP and our NADPH in both of these processes, understand what they do, and understand where those reactants and where those products are coming from and what purpose they serve. So I will catch you guys in the next one when we are going to look at what happens to all that sugar energy that we produce from photosynthesis and how we use it to create cellular energy in the process of cellular respiration. And on that note, have a great one, guys. I'll catch you later.